Good afternoon.
Good afternoon, sir.
Good afternoon, sir.
Good afternoon, sir.
Okay. So in this week we started with the antibiotics chapter, and in our first two lectures on antibiotics, we completed the introduction part, and we started with the beta-lactam antibiotics. So today we'll discuss on uh, mainly the stability of beta-lactam antibiotics, how they are stable in acidic environment, and how they are stable in beta lactamase enzyme environment. And then we'll study the structural activity relationship of this class of compound. And yesterday we discussed on the mechanism of action and various classes under the beta lactam antibiotics like penicillin, cephalosporin, carbapenem, monolactam. That part we covered. So at the end of this week, we'll complete this first part or A part under the antibiotics. And there are these 6 points, but the first three are important. Our first three are the major parts: aminoglycosides and tetracycline, while macrolides, lincomycin, and polypeptides are the smaller units compared to the first. So for all these 6 units, we try to cover the SAR, mechanism of action, synthesis of the compounds, nomenclature, brand names, and the side effects. Few of the things that you can also find out from the literature, like the brand names and the nomenclature for the important compounds. Okay, so these are the few slides uh, that we already studied in last two lectures. Mainly we covered the definition, history of antibiotics, then timeline of the antibiotics, when which antibiotic was discovered, classification of antibiotics based on their uses, based on their mechanism of action, and based on their chemical structures. Okay, these are the three ways we can classify antibiotics. For medicinal chemistry, you should write the classification based on their chemical structure, and that is this one. That is antibiotics under beta-lactam class, aminoglycosides, tetracycline, macrolides, lincomycin, and polypeptides. So the beta-lactam again classified into the penicillin, cephalosporin, monolactam, and carbapenem, because all these four consist of beta-lactam as a common heterocycle. Okay, these are the structures of representative structures of each class of the compounds, like penicillin, cephalosporin are from beta-lactam. Then tetracycline is having these 4 cyclic rings. It is itself a name of class as well as the drug name, tetracycline. Macrolides example is azithromycin, having a multiple carbon cyclic chain. Lincomycin having these specific structural features. And last one is a polypeptide, where the multiple peptide rings or units are attached to each other. So this is about is just brief information about how this different chemical class looks like. So we started with the beta-lactam, and very first we understand these two important compound under the beta-lactam, that is penicillin and cephalosporin, because they are clinically used widely compared to the monolactam and the carbapenem. So you also seen the various formulations of the penicillin, like injections, capsules. Cephalosporin is also available in the various formulation forms. Mainly these drugs are given intravenous because they are very labile to the acidic environment. Why? Because you can see here the presence of amide linkage as well as this cyclic amide, and both are highly unstable in the acidic environment. So if you take them orally, in our stomach, the pH is very acidic, maybe 2, 3, depending upon the food and absence of food. And this acidic, in that acidic pH, these compounds are highly unstable. So generally they prescribed or they are available in the intravenous formulations. Okay, these are the compounds under the beta-lactam, penicillin, cephalosporin, monolactam, carbapenem. And red color ring is beta-lactam, and which is common in all the compounds. Okay, we also understood the numbering, and which is very important for today's lecture, because we will discuss the SAR, that is structural activity relationship, and it is depends upon the numbering pattern. So there are two ways we can number the penicillin or the beta-lactam rings. First is the USP numbering, that is United States Pharmacopoeia, and it starts from the nitrogen, which is linked in between beta-lactam and thiazolidine ring. So it starts from this nitrogen and it goes anti-clockwise. That means after one, it goes to this carbon, which is attached to the carboxyl. Then third carbon having the 2-methyl group attached to it. Fourth carbon is a sulfur group, also called as a thia group. Fifth is a carbon which doesn't have any substitution. Fifth is linked to the beta-lactam. Sixth is a position of this class of compound which is can be substituted with the different groups, okay, through amide linkage. So here is the possibility that you can vary the structure and you can check the improvement or decrease in biological activity. You can also study the stability of this substitution. You can also study the effect of this substitution on the stability of the compounds. So that part we'll study today in the structural activity relationship. But for that, it is very important to clearly understand the numbering pattern of this beta-lactam class of compounds. Another way of numbering is a CAS numbering, called as a chemical abstract numbering, and it is start from this sulfur. So one number is at the sulfur, then two number is at this carbon which is attached to the dimethyl, three number to the carbon which is attached to the carboxylic, fourth number to this nitrogen, and then to the carbonyl like this. So we'll prefer the USP numbering and we'll study according to the USP number because it is given in the references book. Okay, we can also cut down this structure into the different units. The pink color is showing the presence of beta-lactam. You can also see that beta-lactam consists of two chiral centers, and that is why this bond between the carbon number 5 and the nitrogen is towards the observer, or it is a beta bond, and so the ring is called as a beta-lactam. If we call it as alpha-lactam, then the bond is away from the observer, so it is denoted by the dash lining. Another chiral center is at position number 6, and here also the substitution is towards the observer. So it is a dark line showing if this NH is away from the observer, it will be shown as a dotted lines. So these are the two chiral centers which is in this pink colored ring or the beta-lactam ring. Another unit which is attached to this is a thiazolidine ring. And this thiazolidine ring is connected to the beta-lactam ring. And you can see here at the position number 2, also the chiral center and this carboxylic unit is away from the observer. That means it is at the back plane, at the back side of the plane of this beta-lactam attached to the thiazolidine ring. Okay, and so this is amide linkage, carboxylic unit, and R. R is a variable, so it changes depending upon the structure to structure. You can also study this beta-lactam in consisting of these different units shown on the figure, shown on this slide. So there are different ways of the presentation. We also study the IUPAC name of this compound, one of the representative compound called as the benzyl penicillin. So penicillin is a common word, which is common for all the penicillins under this class. Only the variation is at this R position. So if it is a benzene substitution, it is called as the benzyl penicillin. If it is a phenoxy substitution, then it is called as the phenoxymethyl or the phenoxy substitution or phenoxypenicillin. So depending upon this R group, there are different namings for these penicillins. So here it is shown how these penicillins can be named by chemical names, then can be named by their generic or the common names by which the different drug formulations are available, and there are substitutions. Okay. So for example, just now in the previous slide we studied there is a benzyl penicillin where the benzyl group is attached as at the R position. It is also called as a penicillin G. If it is a phenoxymethyl penicillin, then it is called as a penicillin V. Okay. Amoxypenicillin having the amino hydroxybenzyl penicillin. So like at this methyl position, which is attached in between amide and benzyl ring, there is a NH2 substitution. So why the NH2 substitution, why the carboxylic substitution at this particular methyl group, that we'll study today in the structural activity relationship. We also understand the mechanism of action of this drug, and we simply, the simple way of understanding the mechanism is that beta-lactam class of compounds break down the networking between these peptidoglycan linkages. Beta-lactam class of compound break down the linkages between the peptidoglycans. So what is peptidoglycans? Peptidoglycans are a mesh-like structure between two phospholipid chains. So these two phospholipid chains having the peptidoglycan linkages or network. And what is the role of this peptidoglycan linkage? It will give the rigidity to the cell. And that is very important for the cell wall to remain in its intact form. If this linkages break down, then peptidoglycan will no more gives the rigidity and the cell becomes leaky. Okay, cell becomes fragile and it will be get breakdown, and final result will be the cell lysis. So this is what they explain in this flow chart also. Beta-lactam antibiotic first bind to the penicillin binding protein called as a PBP. Then they enters into the cell membrane, mainly at the peptidoglycan, and they break down the linkages, and thus the cell undergoes lysis. This is the mechanism of action for the beta-lactam or the penicillin, cephalosporin, and other drugs. This is another way of representation that how these linkages are there between the peptidoglycan. So it is linked through the amino acid chains. So here exactly the penicillin act, it break down the amino acid linkage. And how it is break down the amino acid linkage? By inhibiting the enzymes called as a transpeptidase. So transpeptidase is the important enzyme which helps in the formation of linkages. So these enzymes get acylated and thus they becomes inactivated. So this is the clear mechanism for the penicillin class of compounds. Okay, so this is the part we covered in the last lecture, and today we'll start with the stability. Any questions?
No, sir.
So as we seen the structure of beta-lactam antibiotics, we understood that there is a 4-membered ring called as a beta-lactam, and there is a 5-membered ring called as a thiazolidine, and also there is a amide linkage. So if we observe the structure in a chemical point of view, the stability point of view, there are many positions which can be easily break down in a harsh condition. Harsh condition means if we add this compound in a strong acid, that compound can be easily break down because the amide bond can be undergoes hydrolysis. acid hydrolysis or base hydrolysis. So they are labile compounds. They are not stable compounds. Okay, so this is the one disadvantage or the drawback of this chemical class of the compound. Though they are highly potent, but they have the stability issues. Another way of this compound can be chemically break down is by beta lactamase enzymes. So what is beta lactamase enzyme? As the name suggests, the beta group or the beta position of the beta lactam ring is can be break down by which enzymes? Lactamase enzymes. So they, as we know that there are different enzymes like esterase. Esterase can break down the ester bond. Amidase enzyme can break down the amide bond. Likewise, the lactamase enzymes can break down the lactam ring. And at which position it breaks the lactam ring? At the beta position. So they called as a beta lactamase. And where these enzymes are present, these enzymes are not present in the human body. Okay, they are produced by the bacteria. So it is interesting thing that bacteria produce these enzymes in response to the antibiotics. And this enzyme, once produced by the bacteria, this enzyme will break down the antibiotics and make the antibiotic ineffective. Are you getting me? So see one example. So this is one of the penicillin, and if this penicillin is exposed to the beta lactamase enzyme, which is produced by the microorganisms, then the beta lactamase enzyme break down the penicillin ring. You see here, the beta lactam ring get open up. This ring will get open up, and once it is open up, the drug will become inactive. Same case in case of the cephalosporin, also consists of beta lactam ring, and when it is exposed to the beta lactamase, which is produced by the microorganisms, this enzyme will open up the beta lactam ring, and once it is open up, the drug become inactive. So the microorganisms are very smart, and they produces beta lactamase enzyme to make the drug ineffective. What is this called as? What this behavior of the microorganism is also named as developing dash against the drug. Okay, as we make the drug to kill microorganisms, microorganism also develop some strategy to make the drug ineffective. What is it called as?
Resistance.
Resistance. Very good. So this mechanism of producing beta lactamase enzyme by microorganisms, and that enzyme make the drug ineffective. This whole process of making the drug ineffective is called as a resistance. So this is how the bacteria develop the resistance against beta lactam antibiotics. Okay. So it is not like if you give the penicillin and penicillin will kill all the microorganisms. This will not possible because bacteria also have their own mechanism to protect oneself, and this mechanism is the resistance mechanism. So penicillin G, penicillin V is sometimes is completely ineffective against the microorganisms, and this is because of this particular effect. So how to tackle this situation? We require to modify the existing antibiotics. We need to modify the penicillin structures in a such a way that it will be not deactivated by the beta lactamase enzymes. Okay. So there are in a structure, if you observe the structure, there is a position R can be modified. Okay, so you have all the scope to modify the R position in a such a way that the drug will not bind to the beta lactamase enzymes. And if it is not bind to the beta lactamase enzyme, then beta lactamase enzyme cannot break down the drug. So this is the one strategy that developing the drug structure. Okay, so this is the main issue. Another stability problem of penicillin and the other beta lactam antibiotic is the acid stability. Okay, see when because this bond is labile, even this both the amide bond, you take the amide bond of the beta lactam which is inside the ring, or you can take the amide bond at the position number 6 which is outside the ring. Both the amide linkages are acid labile. That means they can be break down in the highly acidic conditions. So if we give this penicillin G orally in form of a tablet or capsule or oral liquids, this active constituent will finally reach to the stomach, and pH of stomach is acidic, maybe 1 to 2 in highly acidic situation, maybe 2 to 4 in a whenever you take a food. So at any situation, the pH of stomach is acidic and this bond can be easily break down in those acidic environment. And the final result is that you will not have your intact penicillin structure in the body. If it is break down, then it will be no more active. So there is also this pharmacokinetic issues or stability issues of the penicillin if you take it orally. Okay. Also, there are certain sites in the body have the different pH. Okay, like in the lysosome of the cell, having the pH 4 to 6. There also this compound can be break down. Okay, so these are the main stability problems of the penicillin compound, and we will see the how we can make the compounds acid stable, or we can make the compounds uh, stable against the beta lactamase. That is the main focus of our understanding of the structural activity relationship. Okay, we'll come back to this slide, uh, of this slide particularly after we study the SAR. Okay, so before studying SAR, we'll again go back to the different structures of the penicillin, just to understand which is the common structural features and which is a variation in the structure. So this is the common scaffold of the beta lactam antibiotics or the penicillin class of compound. And where is the variation? Variation is only at R position. Remaining all the parts are the same for this all the penicillin structures. Okay, so only R part is different. So this R may be the phenyl ring attached to the CH2. This R may be phenyl ring attached to the CH2 through oxygen. This R may be phenyl ring attached to the CH attached to the NH2. Even the phenyl ring can be substituted with hydroxyl units. Even the phenyl ring can be substituted with the dimethoxy units. So based on this, there are different names like penicillin G, penicillin V, ampicillin, amoxicillin, methicillin. You must have heard this word, amoxicillin injection or ampicillin injection. Okay, penicillin V injections or saline bottles containing the penicillin. We can actually inject the penicillin V into the saline bottle, and through the saline we can transfer this drug into the patient's body. So these are familiar compounds and these are their chemical structures. More important thing to understand that which is a common thing and which is a variation in the structure. Okay. Now we start our actual point, that is the SAR. So first you go through these points, you read this slide, try to understand what it's saying at each position, and then we discuss it. Done?
Yes, sir.
So the SAR is explained systematically, considering the each position of the beta lactam ring. All of you are now familiar with the numbering system. So we'll start with the position number 1. Okay, as one important point before starting the SAR is that as this is a common scaffold in all the penicillins, that gives us the clue that these all the features are important. Because if these are some of these are not important, then this can be varied. Okay, this can be removed off and still we get the active compound. But as there is nothing is changed in all the penicillins, that means each and every substitution is have some effect in the activity. So see here at position number 1, the nitrogen is important for activity. So you cannot replace the nitrogen with sulfur or oxygen or simply with the CH or with any other heteroatoms. So this nitrogen has some structural feature that will help this drug to kill the microorganisms, to break down the peptidoglycan linkage. Position number 2 is having a carboxylic group attached to the carbon, and it should be free carboxylic group. It should not be like CO, CH3 or CH2OH or any other ester group. Okay, like C double bond OCH3. This simple substitution or small substitution will also not tolerate for this class of compound to show its original activity. Because if you replace COOH with, for example, COOCH3, then also activity reduces. If you replace COOH with the CH2OH, if you make carboxylate to the alcohol or ester, it also reduces the activity. That means the plain carboxylic group is important. Position number 3, where the small alkyl substitutions are important. Which are the smallest alkyl substitution? Is the methyl. So methyl, 2 methyls are attached to it and both are important. If you remove one of it, also activity reduces. If you remove both of, then also activity reduces. If you replace methyl with ethyl or propyl or butyl or isopropyl or benzene or any heteroatom, then also it is observed that the activity of the compound is reduces. But it indicates it only requires the small alkyl substitution. Position number 4, where you can make the sulfones instead of sulfur, or you can also make the disulfo or the sulfoxide instead of sulfur. But see, if you do that, then what happens? There is definitely the stability of the penicillin will improve. Okay, and which stability? Mainly the beta lactamase stability, because it is away from the amide linkage. Okay, you can see here both the amides are quite away from the sulfur group. So this sulfur change to the sulfone and sulfoxide will improve the beta lactamase stability. But on the other side, there is some decrease in biological activity. Okay, so you have to compromise with the biological activity if you want to get the stability against beta lactamase. So that is possible by changing the sulfur position. So do you understand these 4 points?
Yes, sir.
Now, these are the 2 important points, and these are more related to the now beta lactam ring. Initially, the first 4 points are to the thiazolidine ring. Now it is to the beta lactam. So beta lactam having mainly these 2 separate positions, position number 6 and position number 7. And there is no substitution at position number 5, so it is not included in the SAR. So we directly jump on the position number 7 before 6, because the uh, position number 6 having the multiple points to discuss. So we'll directly start with the position number 7 first. So 7 is having a carbonyl group, plain carbonyl group, and which is in the beta lactam ring. And this carbonyl group is essential for activity. So if you reduce this carbonyl to the methyl, then also there is a loss of activity. If you replace this carbonyl with another substitution like CHOH or CH methyl, any other, then also you will find the decrease in activity. So what is important? Plain carbonyl group. Okay. Now here is the multiple points to know, and that actually important for the SAR. So you have only scope at position number 6 to vary, or you have the scope to vary at the position number 4, which is the sulfur. So only these two positions can be changed in the structure. Remaining any other position you cannot change, or you cannot touch only to make the derivatives, because if you do that, then activity will lose. So what changes you can do at position number 6? So here also you should attach any substitution through amide linkage. You cannot attach any substitution through the you cannot attach any substitution directly to the position number 6, or you cannot attach direct any substitution to the any other linkage like ether linkage or ester linkage. That will not be tolerated. You have to attach through the amide linkage only. So there is only way to attach different groups at position to the carbonyl groups. And which substitutions are generally favored? The electron withdrawing substitutions. So electron withdrawing group can be attached to this carbonyl carbon. Okay. So why this electron withdrawing group? Because it will make the compound more acid stable. Okay. So our final aim is that to increase the activity of compound and to make the compound stable. Already the penicillin have the good activity. So main focus is of derivatization is to increase the stability. So we should attach the R group in such a way that it will make the compound acid stable and it will make the compound stable against the beta lactamase. That is only two goals in the substitution selection for this class of compound. So electron withdrawing group make the compound acid stable. Why? The reason is that when you attach electron withdrawing group, it will make the carbonyl group less nucleophilic. Okay. So any bulky groups here, okay, and this is the one reason why we can attach the electron withdrawing group here. So as you attach the many substitution on the phenyl ring like hydroxyl group or chlorine group or bromine group, that make the phenyl ring again more and more electron withdrawing, and that will give the more stability to the compounds. Okay, this is the one way to make the compound acid stable. Another is that how we can make the compound stable against beta lactamase. For that, the important thing is you can attach here the bulkier groups. So there are many electron withdrawing groups like chlorine, fluorine, bromine, iodine, sulfonyl, carbonyl, but that will not help because though they are electron withdrawing, they are not bulkier. So we want both the things. We want acid stability also, and we want the stability against beta lactamase. So we need to have both the characteristics. Which characteristics? We should have the electron withdrawing as well as the bulkier groups. Because electron withdrawing protect against the acid stability and bulkier groups will protect against the beta lactamase. Okay. How the bulkier groups protect against the beta lactamase? due to steric hindrances. If you attach here the bulkier groups like phenyl, phenyl methyl, phenyl methoxy, or CH attached to the phenyl, which can be substituted with NH2 or NH with more substitutions, then these make the position more bulkier. What is mean by bulkier? It will produce the steric hindrances, because what will happen finally, this molecule will go and bind to the beta lactamase enzymes. So enzymes is a protein, there is an active site, and in that active site this enzyme, this compound will bind, and then it will be breakdown. Now if you attach some bulkier or steric group at R position, the compound will not fit or bind to the enzyme. And if it is not bind to the enzyme, then enzyme cannot recognize it, enzyme cannot break down it. That is the logic to attach the bulkier group at position R. Okay. So this is how the position 6 can be varied, either with electron withdrawing or by the bulkier groups, to make the compound stable. So this is all about the SAR. You can also understand SAR based on this image. One more point which is not mentioned in the previous slide, that at position number 5 and 6, it should be cis stereochemistry. That means both positions substitution of H atom should be at the one on the one plane. It should not be trans, because trans is giving the position of H at the opposite ends or at the different planes. Okay, cis means both the hydrogens are at the same plane, and that is requirement for the good activity. So that is one more point you can add, and all other points that we already discussed. Okay. So this is about the SAR of this class of compound. Now we'll go back to our slide, which we intentionally will discuss later. That is the okay. See now, these are the few examples and how they are modified to make the compound stable. See, penicillin G. This is the structure for penicillin G. What is penicillin G? It is a benzyl group. Benzyl means benzene with CH2. It is benzyl. So if you attach benzyl, the compound is acid labile. That means the carbonyl group can be easily attacked by the hydroxyl units, and the hydrolysis will occur here, and the compound will no longer will remain as it is, and it will become unstable. Similarly, at beta lactam also it can be breakdown. So if you see the chemistry of hydrolysis, see here, this H3O having the H plus and OH minus, and this make the hydrolysis of esters as well as amides. So whenever the hydrolysis occur of amide, it produces the free carboxylic group and free amine group. Okay, so either of the free amide group or the amide which is inside the ring. So wherever the hydrolysis occur, it produces the free carboxylic and free amine. So this hydrolysis process depends upon the electron density at this carbon position. So if we change this electron density, we can actually control the acid stability. So benzyl is not providing sufficient electron environment here, and so that it can be easily attacked by the hydroxyl units. But if you attach some electron withdrawing group like oxygen instead of a methyl, then oxygen will withdraw the electrons and it make the compound acid resistance. So it cannot be easily break down by the acid hydrolysis process. And so that penicillin G cannot be given orally, but penicillin V can be given orally, because here the electron withdrawing group is attached at position R. Here there is no electron withdrawing group, and that clearly gives the difference that penicillin G is orally inactive, but penicillin V is orally active. So this is how we can modify the structure if we understand the structural activity relationship. Another example is this ampicillin. See the ampicillin where the, okay, these all are acids. Okay, these are all are compounds which are stable in the acidic environment. Even ampicillin, amoxicillin also having the NH2 group near to the carbonyl, which is electron withdrawing, and so they make the compound acid stable or acid resistance. You can use any terminology, acid resistance or acid stable. That both are indicating that the compound is stable in the acidic environment. So penicillin B, ampicillin, amoxicillin are the acid stable compounds, while penicillin G is acid unstable. Okay, this understanding is important because in objective type question may be asked why the penicillin G is acid unstable, or which of the following is acid stable and which of the following is acid unstable. So you should be clear with this understanding. Okay, any questions? Anything that you are not understood? Clear with all the points?
Yes, sir.
Yes, sir.
Good. So here we almost completed the beta lactam antibiotics. There is 1-2 slides for the cephalosporin and other beta lactams because remaining points are same like of the penicillin. Just we'll see the few structures of the cephalosporin in the next lecture, and we'll start with the tetracycline. If you have any questions, you can ask me, or you can read from the book, and if you get any query, also you can ask me in the next lecture. So all of you refer Wilson and Griswold antibiotic chapter. You read all the introduction part that will help you understand the next points. And you can also start preparing your own notes on this chapter. Okay. So you can use the points from the slide and you can write your own text. So generally the question asked on this part is that write a note on beta lactam antibiotics, or give in brief about the stability of the beta lactam antibiotics, or write in detail the SAR of this penicillin, or generally they asked explain the chemistry and mechanism of action. In that all the points get covered. If they say chemistry, then it comes the structures, comes the SAR, then numbering patterns, stability, all things comes under that. Mechanism of action generally focused in the pharmacology syllabus, but here also the mechanism can be asked, where you can at least should write at which position, at which exact point the beta lactam antibiotics act and kills the microorganisms. In the classification of beta lactamase, you should classify the beta lactamase in 4 classes: penicillin, cephalosporin, monolactam and carbapenem, and under each class you should write at least 3-4 examples with structure. Okay, like for the penicillin, few examples like penicillin G, penicillin V, ampicillin, amoxicillin, and then you can write a common structure as one, and then just write the structure for the R. Okay, so these are the type of questions can be asked in exam. Anything else you want to discuss? No? Okay then.
No, sir.
Good. So tomorrow is our tutorial lecture. So at 1:30 tomorrow, all of you uh, must have prepared the classification of those 4 class of compounds that we discussed in our first 2 lectures, that for which compounds you have to prepare the classification. So what you have to do is that I will prepare one subjective question on ERP, and you just upload your notes of classification on ERP. I will check on ERP. I will assign the marks to your notes. Okay.
Yes, sir.
I will create one subjective question for classification of drugs for antimalarial, sulfonamide, urinary tract infection, and antiviral. Under that notes you upload, under that question you upload your notes. Tomorrow at 1:30. So I will stop.